Oh,
Yeah.
sorry. Okay, great. Welcome everyone to our Fellow Science session, where we'll be hearing from our distinguished fellows and about their research related to communication between cells and beyond. My name is Rachel Zwick. I'm a postdoc at UCSF, and I'll be moderating this session today with my colleague Aishwarya Iyengar, and I'll have her introduce herself now too.
Hi, I'm a postdoc at Brandeis University in Waltham, MA, and I'm happy to be here, and all the best to the speakers.
Thank you. Great. We are all very excited to hear their talks. And we really also appreciate all of the audience being here to support this fantastic community. So for this session, each fellow's presentation will be 15 minutes, and then we will follow with 5 minutes of questions and answers. So at any time during their presentation, please post your questions into the Q&A chat box on your screen. And since time is a bit limited, we might not be able to get to everyone, so please try to keep your questions somewhat pithy so we can be sure to cover as many as possible. And yeah, I think without further ado, let's get started. We'll be hearing from Dr. Anda Kurila, who is a neuroscientist at Harvard Medical School studying somatosensory signals in the spinal cord.
Thank you. Thank you, everyone, and especially to the organizers for this fantastic symposium. I'm a neurophysiologist interested in central representations of touch and pain, and I'm excited to share with you my work in the lab of Dr. David Ginty, where we study subcortical organization of the somatosensory system. Among the senses, touch lies at the heart of our experience of the rich physical world around us. It enables us to perceive a gentle breeze, physical contact of loved ones, as well as the fine textural details and shapes of objects that we hold in our hands. And touch perception is profoundly modulated by context and internal states, and can be altered in disease states. So for example, gentle touch is perceived as painful in neuropathic pain conditions, physical contact as aversive in individuals with ASD, and pain signals can be completely overlooked in certain arousal states. So for example, wounded soldiers on the battlefield do not experience significant pain. Mirroring the complexities of touch perception, there are multiple ascending pathways for light touch from skin to cortex. And this include the direct dorsal column pathway that carries signals from the fast-conducting low-threshold mechanoreceptors, or LPMRs, directly from skin to brainstem, and from there somatosensory information is then relayed via the thalamus all the way to the somatosensory cortex. But this is not the end of the story. In fact, the majority of the A beta LPMR synapses reside in the spinal cord and not in the brainstem. And all cutaneous and from here, projection neurons, the postsynaptic dorsal column projection neurons also carry mechanosensory information to the brainstem to meet with the direct pathway in the brainstem. And this fundamental circuit is conserved from mice to primates. And while it makes sense that we have the direct pathway, this is a quick, fast way to get sensory information from skin to cortex. The role of the indirect pathway is completely mysterious, and I'm really fascinated by this question. And so in order to understand what the role of the indirect pathway is in somatosensation, we need to be able to understand how touch is encoded in the spinal cord dorsal horn. So when I joined the Ginty lab, these were the outstanding fundamental questions that I really wanted to answer. So I wanted to know how mechanosensory signals are encoded in the spinal cord dorsal horn, how the rich mechanosensory input from the periphery is functionally organized to give rise to local circuit dynamics, and then what are the response properties of the projection neurons that form the indirect dorsal column pathway, and lastly, what does spinal cord processing shape And so really answering these questions would reveal the role of the PSDC indirect pathway in touch processing. And to answer these questions, our general approach is to use mouse molecular genetic strategies to access the neuronal components of the somatosensory periphery and of the spinal cord. But of course, genetic tools are not enough. We also need to be able to record sensory evoked activity from the living spinal cord. And so to this end, I have developed a preparation for large-scale electrophysiology in anesthetized mice that allows me to record simultaneously the activity from many lumbar dorsal core neurons, extracellular action potentials, while delivering precise mechanical stimuli to glabrous skin. And we deliver mechanical indentations of increasing intensities, and we can deliver these also on a grid to really map cutaneous receptive fields of second-order dorsal core neurons. And so when we use the range of well-controlled mechanical stimuli, we found that the most of the dorsal core neurons that we recorded from responded to light touch, with a broad range of functional response profiles. And so we wanted to know what the extent of this functional diversity is in the spinal cord, and we performed unsupervised clustering on our large data set of more than 5,000 single unit responses. And this analysis has divided our spinal cord recordings into 6 functional principal groups that you can see here. And neurons across these 6 functional clusters differ in terms of their sensitivity to step indentation. So for example, units in functional cluster 3 are some of the most sensitive units that we recorded from. A lot of them respond to 1 millinewton force. And then we also have units that have higher force thresholds, like those in cluster 2 and 6. And units in cluster 1 have transient responses to step indentation. So they really only respond at the onset and have a small off response. We also have units in cluster 4 and 5 that have sustained responses to these step indentations. And across these 6 physiological response profiles, dorsal horn neurons also had varied receptive field sizes and shapes, and they encoded a mechanical vibration with distinct response properties. And in addition to the remarkable diversity of mechanoreceptor input in the spinal cord, the spinal cord also harbors a diversity of local interneurons, both excitatory and inhibitory subtypes. Here are 2 major genetic populations, the parvalbumin expressing interneurons and ROR beta expressing interneurons, shown here in green. And we really wanted to know whether these 5 physiological response profiles can be mapped onto genetically known populations. And so to answer this question, we use our spinal cord specific molecular genetic toolkit in combination with optotagging strategy. So this really allows us to find distinct dorsal horn neurons in our extracellular recordings and map their responses to light touch. And when we do this, we find that ROR beta neurons have transient responses to step indentation with little off response at the, during low mechanical forces. And based on their response properties, they map predominantly onto functional cluster 1. In contrast, parvalbumin expressing interneurons have sustained responses and prominent off responses to the step indentations and are very sensitive. And based on their response profiles, they map predominantly onto functional cluster 3. And we've used the sampling of 5 distinct excitatory and inhibitory interneurons in the spinal cord. And this analysis really allowed us to conclude that we can map genetically known populations on our 6 functional response profiles. We also wanted to know how these 6 response profiles are generated. And from primary sensory neurons, loss of function and gain of function manipulations, we find that all of our 6 functional clusters receive convergent input from many different LTMR subtypes, as well as high threshold mechanoreceptors from the skin. So we find this extensive convergence of mechanosensory input that gives rise to these functional response profiles. Next, we wanted to know how the diverse dorsal horn network might influence the responses of these functional response for all of our cluster types. And so in order to answer this, we used ROR beta and PV neurons as genetic entry points into manipulating cluster 1 and cluster 3 because they map onto these distinct clusters. And this allows us to ask what their role is in shaping the functional response type of the other dorsal horn neurons, and ultimately shaping the output that is the indirect dorsal column pathway. And in addition to their diverse responses to step indentations, RR beta and PV neurons also participate in distinct synaptic inhibitory circuit motifs in the spinal cord. We find that RR beta mediates sensory evoked feed-forward inhibition, while PV neurons participate in presynaptic inhibitory circuits, forming axoaxonic synapses directly onto the primary sensory neuronal terminals. And we can use genetic inactivation strategies to delete GABA receptors selectively in all somatosensory neurons, and this in part eliminate RR beta mediated presynaptic inhibition. PV-mediated presynaptic inhibition, and we can also genetically inactivate signaling from the RR beta neurons, and thus manipulating functional cluster 1 and functional cluster 3. We can now ask how does the interconnected dorsal horn network influence the responses of the output neurons of the PSDCs. And so here we're recording selectively from the output neurons. And what we find is that as the population, they have very diverse response properties to these very basic step indentations. So you can appreciate that some of the PSDC neurons are more sensitive and have more sustained responses, whereas others have more transient response profiles. And so this is in contrast to the genetically labeled dorsal horn interneurons that had stereotypes responses to this simple tactile stimuli. And we find that in the absence of feed-forward or presynaptic inhibitory circuit motifs in the dorsal horn, this functional diversity at the level of the outputs collapses. So now all PSDCs are more sensitive and they have pronounced sustained responses. And we find that dorsal horn neuron also shape the receptive fields of the PSDCs. In the absence of feed-forward or presynaptic inhibition, we see an enlargement of receptive fields in PSDC neurons. So really the heterogeneity of the PSDC responses depends on these dorsal horn inhibitory circuit motifs. Next, so we find that the PSDC responses really depend on the inhibitory components of the deep dorsal horn circuitry. And so we wanted to know whether these differences that we see may influence the responses at higher levels of the somatosensory cortex, at higher levels of the somatosensory hierarchy. And so next we recorded in somatosensory cortex in awake mice, while giving the same step indentation to glabrous hindpaw. And what we find, as previously reported, is that units in somatosensory cortex have transient responses to these step indentations. Now, when we selectively disrupt spinal cord mediated feed-forward inhibition, we find that their sensitivity and their spiking at the onset and during the offset of the step indentation is enhanced. And this is what we would predict from the responses in the PSDC output neurons. So mechanosensory processing in the spinal cord can really shape touch evoke responses in the brain. And what was even more dramatic was when we mapped brush evoke receptive field of units in somatosensory cortex. So in the absence of spinal cord feed-forward inhibition, we find that the receptive fields of units in somatosensory cortex are dramatically enlarged, really highlighting the role of the indirect dorsal column pathway in shaping how light touch of the skin is represented in the brain. And so in summary, what I've shared with you so far is that using large-scale electrophysiology in the spinal cord, we find 6 principal functional response profiles, and dorsal horn interneurons across these functional clusters receive either direct or indirect input from many different mechanoreceptor types. We find that the outputs of the dorsal horn, the postsynaptic dorsal column projection neurons, have a diversity of responses to light touch stimuli, and this functional diversity collapses in the absence of distinct inhibitory circuits in the spinal cord. And we find that these alterations are also reflected in somatosensory cortex. So this highly interconnected dorsal horn network enables dynamic flexible tuning of the PSDC output neurons and really dictates how neurons in the brain respond to touch. And we're very excited about this work because we think that this really brings us closer in understanding what the role of the indirect pathway is in touch processing. So while the direct pathway carries unmodified mechanoreceptor signals from skin to brain, what the indirect pathway is doing is it adds flexibility into the system by reflecting highly convergent input from many different mechanoreceptor types. It broadens the dynamic range of the direct pathway. It allows for flexible encoding of somatosensory stimuli. And we think that this provides a rich substrate that can be recruited by sensory experience and internal states. And I'm very excited to test these directions in my own lab. And the goal of my research program will be to functionally define the subcortical organization of the mammalian somatosensory system and use this knowledge to reveal how internal states and sensory experience can flexibly shape our sense of touch and pain. Building on my postdoctoral work, first I will establish how diverse somatosensory stimuli, so not just mechanosensory stimuli, but also noxious, temperature, and chemical stimuli, are encoded across distinct spinal cord neurons for output to the brain. I will then use this knowledge to ask how internal states and sensory experience can flexibly shape our sense of touch and pain. And long-term and more ambitious goals include using large-scale electrophysiology in awake behaving mice to ask how the dorsal horn encodes more naturalistic behaviors. And so after establishing how the spinal cord encodes a broad range of somatosensory stimuli, we can ask how internal states and disease states may shape subcortical somatosensory processing. And I think we can all appreciate that touch can be profoundly modulated by internal states and altered in disease states. And the spinal cord is really a key locus of this function in a range of somatosensory processing disorders, from ASD all the way to chronic and inflammatory pain. Moreover, the spinal cord receives strong descending neuromodulatory inputs from the cortex and brainstem, including from multiple brain regions that are implicated in the control of internal states, further suggesting that the spinal cord contributes to state-dependent modulation of sensation. So by exploring the spinal cord as a rich and functionally complex first site of central nervous system processing, I can ask how top-down signaling that encodes internal states combines with bottom-up sensory input to really shape our experience of the rich physical world around us. And with that, I would like to thank first and foremost my postdoctoral mentor, Dr. David Ginty, everyone from the Ginty Lab who contributed to this work, my undergraduate students that I had the privilege to work with, our collaborator Chris Harvey and Si Tsang, our funding sources, and all of you for listening. And I'm excited to hear your questions. Thank you.
Great. Thank you so much, Dr. Krila. That was really interesting. And I think people should start sending their questions in the chat. And we're going to start off from a question from Aishwarya first.
Hi, it was a great talk, Dr. Shukla. I was just wondering, so you were talking about how different modalities change the experience of these somatosensations, these different somatosensations. Do you have any understanding of how age could be a factor, or gender, while you were doing these experiments? Were they age controlled or of a specific gender or anything like that? Have you noticed anything?
Yeah, so I think that's an interesting future direction. So what I can tell you is that in our electrophysiological recordings in the spinal cord, we use both males and females, and we don't see differences in their tactile sensitivity or responses to mechanosensory signals. But I think in under different behavioral states, we would perhaps expect changes in somatosensory encoding between males and females. We haven't looked at aging, but of course, tactile signaling is going to be not as pronounced in older mice. And so for example, we know that already the mechanoreceptors themselves have lose their sensitivity with age. And so I think this is going to feed into central circuits, of course. And so we would expect to see blunted responses to tactile stimuli in older mice.
Thank you so much. Let me just check.
I was very interested in sort of a related question about your future work looking at disease states on somatosensory processing. And I was wondering how much we know about that and what types of diseases are most pertinent for that line of work.
Yeah, so I think, yeah, so I'm very excited about this direction because it seems like the spinal cord is really a locus of dysfunction. Of course, we know for pain processing and for motor disorders, but recently my collaborative work has shown that in neurodevelopmental disorders such as autism spectrum disorders, the spinal cord circuits are hyper sensitive to light touch when we selectively delete ASD-associated genes only in somatosensory neurons. So I think it's very interesting to really study spinal cord circuits because, for example, the two different modes of synaptic inhibition that I study in a non-disease states, it turns out that both the so RR beta and GRBRB3, which is the GABA subunit that we delete to functionally manipulate these distinct circuit motifs, they are actually ASD-associated genes. And so when we eliminate these modes of inhibition in the spinal cord, we see dysregulation of tactile processing, and perhaps this is what underlies this tactile aversiveness that some individuals with ASD report.
That is extremely interesting. Thank you. Great. And I think we have time for about one more question. And I can ask it, or Aishwarya, I just wanted to check in with you. Okay, I'll go ahead. Oh, sorry. Yes.
Yeah. There aren't any questions in the other chat as well. So yeah, you can go ahead, Rachel.
Okay, well, if you don't mind, then I'll just ask another one. Then I was actually wondering if you could talk a little bit more about the spatial distribution of the responses that you mentioned, and if we have any inkling as to how this is organized or how the distribution might vary in different contexts.
Yeah, no, I think that's a really great question. And so we record from lumbar spinal cord that receives direct input from the glabrous hindpaw. And so our responses that we've characterized are all from input from the hindpaw. And we find that dorsal core neurons actually have really large receptive fields in the hindpaw, which is surprising because this is glabrous skin. Glabrous skin is supposed to be important for tactile acuity and tactile discrimination. I think a great direction would be to actually look at these response profiles across different skin types. So, for example, in hairy skin or perhaps forelimbs, and really, see whether perhaps we have dedicated circuits for in the spinal cord for different skin regions.
Wow, fantastic. Okay, great. Well, thank you so much. And we're going to go ahead and move to the next speaker. So that is Dr. Krishna Mudumbi, who is here to tell us about his work studying receptor tyrosine kinases at the Yale Cancer Biology Institute.
Let me get my screen share ready here. All right, thank you so much for that introduction. Yes, my name is Krishna Mudumbi, and I'm a postdoc at Mark Lemon's lab at the Yale School of Medicine. And today I want to talk to you a little bit about how I've been probing the kinetics of receptor signaling and why I think timing is an important dimension to consider when we think of signal transduction. So I'm sure many of you know, of course, that signal transduction is an important process that allows cells to communicate with one another and also receive and process signals from their outside environment and then react accordingly, right? This process controls lots of cellular processes from apoptosis to metabolism, cell growth, et cetera, et cetera. And as such, it's very tightly regulated and typically regulated by receptor proteins. These proteins come in several different flavors that I'm showing you here. So there's channel proteins, there are 7-pass transmembrane proteins like G protein coupled receptors. We have single pass transmembrane proteins, as well as intracellular receptor proteins. But for the purpose of this talk, I'm going to be focused mostly on single-pass transmembrane proteins, and in particular, a group of proteins known as receptor tyrosine kinases, or RTKs. As you can see, there's 58 different members of the RTK family, but the Lemon Lab is particularly focused on the ERB receptor, and in particular on the epidermal growth factor receptor, or EGFR. As you can see, EGFR has a very similar architecture to the other RTK family members. It has an extracellular ligand binding domain, and once a ligand is bound, these receptors form a nice, beautiful heart-shaped dimer that I'm showing you here. There's a single pass transmembrane region. And we also have intracellular kinase domains, which are able to form asymmetric dimers upon activation. And these asymmetric kinase domain interactions allow for the phosphorylation of tyrosine residues on the C-tail of this receptor. And of course, once this tyrosine residue is phosphorylated, we see the signal transduction happening intracellularly by recruiting adapter proteins like GRAB2 and SOS, the activation of RAS and ERK. Now, signaling itself can kind of come in two flavors. One that we know as biased agonism or biased signaling has been very well characterized in G protein coupled receptors. And the basic premise here is that we have a single G protein coupled receptor, a single GPCR, that has multiple different ligands that can bind to the receptor and result in differential downstream signaling outcomes based on the identity of the ligand. So an example of this could be this orange ligand could bind to the receptor. It could lead to, let's say, proliferation. A blue ligand could bind to the exact same receptor, have a different conformational change, and result in, let's say, differentiation. Now, in contrast to this, EGFR and RTKs are typically thought to be more like on-off switches, where if there's no ligand bound to the receptor, it's considered off, but upon ligand binding, the receptor turns on. But this doesn't fully describe RTK signaling, because RTKs, like many GPCRs, and EGFR also exhibits this, it has several different ligands that can activate the receptor. And again, the identity of the ligand determines the downstream signaling outcome. So this on-off phenomenon doesn't really describe this biased signaling that we're seeing with RTKs. And in fact, recent work from a couple of different groups has started to suggest that kinetics likely define the signal bias that we see in RTKs. And in fact, work from the Lemon Lab has shown really nicely that low affinity ligands like epiregulin, when they bind to the receptor, they result in weak dimers. And this is shown as a sustained signaling kinetics at the level of receptor phosphorylation and ERK phosphorylation. And the cell interprets this as a signal for differentiation. In contrast, when the high affinity ligand like EGF binds to the receptor, we see the formation of more strong dimers. This results in transient signal kinetics at the level of both receptor phosphorylation and ERK phosphorylation, and the cell reads this out as a signal for proliferation. So again, this on-off phenomenon, this basic toggle switch, doesn't really explain the biased signal that we're seeing. And in fact, we can start to understand the importance of kinetics and the role they play in determining these biased signals and how they determine downstream signaling outcome. But unfortunately, it's a very understudied aspect of RTK signaling. And this is really the main focus of my future research program. So in my own lab, I plan to study how signal kinetics of transmembrane proteins specify downstream signaling outcomes. And there's going to be basically two major arms to this approach. The first is to look at receptor activation kinetics. So to try and understand dimer duration lifetimes, how those lifetimes are altered based on the ligand that's activating the receptor or different activating mutations. Another aspect would be to look at the recruitment of downstream signaling kinetics by looking at the recruitment of adapter proteins, phosphorylation lifetime of the receptor itself, look at the activation time for ERK and RAS. And as you can imagine, we can start to piece together this kinetic picture of activation at the level of downstream signaling and at the level of the receptor itself, and start to understand the kinetics that define these cellular outcomes. So what are the kinetics that define proliferative signals versus ones that lead to differentiation? And once we get this kinetic understanding, we can start to create therapeutics that can target these kinetics and alter or bias signaling in a way that's preferential for us. So an example of this would be if we think about cancer cells. They typically tend to be very proliferative. They want to grow and divide. But if we understand the kinetics that control the signal mechanism, we can create therapeutics that target this proliferative signal and instead alter it to a more differentiative signal, and hopefully create new avenues for therapeutics for patients that might lead to better treatment outcomes. The way I plan on answering these questions and asking them is by using my expertise in single molecule imaging and bring that into the field of signal transduction. So as a graduate student, I developed and used various single particle tracking and super resolution light microscopy approaches to look at transmembrane proteins and their distribution on the nuclear envelope, to look at their translocation rates, and then also to look at their transport through single nuclear pore complexes to understand their dynamics in a living cell. So with these tools in hand, I joined Mark Lemon's lab, which is a historically structural biophysics lab. And luckily, Mark had enough funding to let me, in essence, build my own little mini lab within his lab and buy also some fancy microscopes to take this really beautiful structural knowledge that we have of EGFR and start to bring it into a live cell context so we can start to look at the kinetics of receptor signaling, receptor dimerization, and really understand what's happening at the cell surface. So I've had three major projects or areas of focus in the lab. The first one being to try and understand how different domains of EGFR contribute to its dimerization. The second has been to look at how dimer durations determine downstream signaling outcomes by using a single molecule FRET approach that I've developed in the lab. And the third has been to look at receptor oligomerization kinetics and how they affect downstream signaling. Unfortunately, due to time, I won't be able to cover this third topic, but I'm happy to talk offline with anyone that's interested. So the first project, as I mentioned, is to look at how different domains of EGFR contribute to its dimerization. So as I mentioned, the Lemon Lab and many others have given some really great structural context to how EGFR dimerizes, but unfortunately, these images are of course static because they're crystallography structures, and they're also piecemeal because we've only seen how the extracellular domain behaves when it's isolated, and likewise with the kinase domain. But of course, I'm not the first one to try and take this piecemeal image and put it into a live cell context. In fact, in 2010, In-Hee Chung and Ira Mellman used quantum dots, which recently won the Nobel Prize, to tag the EGF receptor, which you're seeing as these black spots, and then track them as they move around on the cell surface. And by looking at the dynamics of receptor diffusion, they noticed that when they didn't stimulate cells with the ligand, these receptors moved quite quickly on the cell surface. But upon stimulation with EGF, there was a reduction in this diffusion coefficient that they measured, so that receptors slowed down by about half. And they used this change in diffusion coefficient as a readout for dimerization and signaling. And in fact, this is what the field has been doing for the last 13 years, but it's actually unclear if simple dimerization alone allows for this change in diffusion coefficient, or if we need the formation of higher order oligomeric structures. And in fact, we also wanted to try and understand how the different domains of EGFR contribute to either this dimerization or oligomerization that we're measuring by this change in diffusion. So I did look at every single domain of the receptor, but for the sake of time, I'll give you the highlights. So just to orient you with what we're looking at here, we're looking at receptor diffusion, so average receptor diffusion per cell. And just like the In-Hee Chung work, we noticed that when we used a wild type EGF receptor in CHO cells, we noticed that when we don't stimulate with EGF, the receptor moves quite quickly on the cell surface. And upon ligand stimulation, the receptor diffusion drops quite significantly. So then we decided to truncate the receptor by removing the kinase domain entirely to see how this affects diffusion. And surprisingly, we found that even though the receptor is still able to form dimers, it's unable to change diffusion in response to addition of EGF. So both without and with EGF, the receptor diffuses quite quickly. Then we also wanted to try and understand how the conformation of the kinase domain might be playing a role. So clearly kinase domain interactions are important from the kinase domain truncation experiment. And then we used an inhibitor that locks the kinase domain into an active conformation. And we noticed that in the active conformation, even without the addition of ligand, the receptor diffuses quite slowly. So then we wanted to see if the slow diffusion can actually be tied back into the formation of higher order clusters. And to do that, we used a super resolution approach known as STORM. And we found a beautiful nearly one-to-one correlation between diffusion coefficients measurements and clustering of the receptor. So if we look at the wild type receptor without ligand, you can see that it doesn't form clusters. However, upon the addition of EGF, we see the formation of clusters, which directly correlates with this fast and slow diffusion that we see. With this kinase domain truncation mutant, we see that clusters don't form even without the addition of ligands. So this fast diffusion that we've measured back here. And then finally, using that inhibitor that locks the kinase into an active conformation, we see the formation of clusters even without the addition of EGF. So again, correlating with that slow diffusional speed very well. So this work is currently on bioRxiv and hopefully should be published soon. But the key takeaway points here is that we noticed that even though dimerization is necessary, dimerization alone is not sufficient for the changes that we observe in diffusion. And in fact, kinase domain interactions and conformations play a necessary role in the slowdown that we observe upon ligand activation. Finally, we also noticed that kinase domain interactions mediate the formations of higher order oligomers. So we can now ascribe a new role to kinase domain interactions that previously wasn't known. So the formation of these higher order oligomers, it was really unclear what mediated that. And we can now clearly show that the kinase domain plays a major role in the formation of these higher order clusters. And this is what we're actually reading out as the slowdown in diffusion. But while this information is really nice and tells us a lot of new information about the receptor and how the different domains are involved, if we go back to this idea of signaling kinetics and how they might be determining downstream signaling outcomes, this SPT measurement is kind of insufficient because it doesn't tell us what dimers are doing. It instead tells us what oligomers are doing. So we need a more direct way of looking at dimerization strengths, which brings me to my second project, which was to really develop this single particle tracking, single molecule thread approach to directly observe dimer durations and how they might be controlling downstream signaling outcomes. So what I did here was I tagged my receptor using a genetic code expansion and bioorthogonal labeling method. So in brief, we substituted our protein, our amino acid of interest, we substituted in an amber stop codon. Then using a tRNA and tRNA synthetase that's specific for this amber stop codon, it can recognize the stop codon and then incorporate an unnatural amino acid that we add to our cell media. And now we have this unnatural amino acid, which we can do some really beautiful click chemistry with, which also won the Nobel Prize, and label with some really bright fret-friendly fluorophores. So in this case, we used Genelia fluors that are really nice and have a really nice fret range. So I'm going to show you an experiment here to give you an idea of what it is that we're doing. So what you're seeing here is single EGF receptors on a cell surface. Green receptors are donor receptors, purple receptors are acceptor receptors, and we can start to track individual receptors as they move around on the cell surface, observe their fluorescence in real time, as well as their spatial location. And what we're going to see is these two receptors are going to come together here in contact and do fret right there. And that's characterized by a change in intensity. So the donor fluorescence goes down, the acceptor fluorescence goes up, and also at the same time, these two receptors are interacting. So we can correlate the spatial interaction of these receptors with their fluorescence and verify that the fret that we're seeing is in fact true, because as you can imagine, live cells are quite noisy. But now with this tool in hand, we can start to try and understand how the different domains of EGFR contribute to its dimerization. Again, using this genetic code expansion method to label the different domains, understand how they're interacting in a live cell setting, and really kind of give life to the static information that we've previously had of EGFR dimerization, and really understand how the receptor more or less breathes in the live cell. We can also try and see how this dimerization lifetime for these different domains can be altered when we use different ligands, and then again, tie this really back into the idea that signaling kinetics or dimerization kinetics are read out by the cell as different signaling outcomes. So as I already said, the directions of my future research program is to use these kinds of tools that I've been developing to decipher the kinetics of transmembrane receptor-mediated signaling. So again, with the single molecule thread approach, the lab will start with probably RTKs, since that's where my expertise lies, but we can easily expand to cytokines or GPCRs, which are very amenable to tagging in this genetic code expansion method. And we can start to understand how different ligands or different mutations or even heterodimerization or heterointeractions alter this dimer duration and how that encodes for signaling outcomes. We can also look at downstream signaling kinetics by looking at phosphorylation order or preference of the receptor. We can look at phosphorylation lifetime, as well as adaptor protein recruitment and stoichiometry. Again, a question that's not only unanswered in the fields of RTKs, but also cytokines and GPCRs. And then another project that I'd love to expand into a little further down the road is to try and look at the dynamics of higher order structures and their formations and the roles that they play in signal transduction and in determining downstream signaling outcomes. So with that, I would love to wrap up and of course, thank Mark, as I mentioned, for more or less providing me all the money that I needed to buy beautiful microscopes, all the members of the Lemon Lab for their contributions to this paper and just intellectually in general, and in particular, a shout out to my undergrads, Daralee Torres, Lucy Kim, and Emma Manji-Capre. The Kate Ferguson Lab, again, for their really great intellectual contributions to my work. And then our collaborators at the University of New Mexico, Diane Lidke and Keith Lidke, with whom we've been collaborating for all these projects. And of course, my funding sources, an early K99 from NCI and an R01 to Mark from the NCI. And with that, I'm happy to take any questions.
Great. Thank you so much. What unbelievable work you're doing. Thank you for sharing with us. I'll get started and everyone can just provide their questions in the chat. So I was wondering, you have an unbelievable level of expertise in choosing super resolution microscopes that I think a lot of us can only dream of using. Can you tell us a little bit about how you choose which technique to use for different questions? It seemed like you had a couple of different approaches.
Yeah, that's a great question. I guess it depends on what you're looking at. So for instance, when we look at cell surface receptors, TURF is pretty amenable to imaging for single particle tracking or super resolution because it gives us a really nice signal to noise ratio when we're looking at cell surface receptor proteins. We could also move, so the work that I was doing as a graduate student was looking at the nuclear pore complex. So we used single point illumination to target single nuclear pore complexes. So something that went more in depth into the cell, but gave us a nice resolution of a single point, much closer to the nuclear envelope. And then I guess, there's this beautiful pyramid that, oh gosh, I'm forgetting his name, and I shouldn't because he's like the father of super resolution microscopy, but he has this beautiful pyramid of the trade-offs during light microscopy imaging, right? So you just have to find what works best for you. If you're really interested in the time domain, you need to find something that'll give you some fast dynamic information. If you need high resolution, you have to sacrifice some things like time resolution and go for more spatial resolution. So yeah,
Great. That's
question.
Very interesting. Thank you. Okay, great. And we're going to take a question now. So EGFR oligomerization, when treated with inhibitor, is very cool. Does the oligomer only affect EGFR homodimerization, or have you looked into heterodimerization with other RTKs in the presence of EGFRI?
No, that's such a great question because oftentimes we're told, oh, this inhibitor is extremely specific for protein X, but they definitely have off-target effects. And it's something I'm very interested in looking at, but we haven't gotten there just yet. But yeah, definitely something we're very interested in looking at.
Great. Great talk. Do you think that the lipid bilayer plays any role in regulating the dimerization kinetics?
Yeah, another really amazing question. So we have a member in the lab, James Hutchinson, who's actually looking at the role of the lipid bilayer in receptor dimerization. Hopefully, before I move on, James and I can work together. So we're thinking about doing a bottom-up approach where once he's done with his assays to figure out which lipids are important for EGFR dimerization, to take those lipids, create an artificial bilayer, supported bilayer, and then look at receptor dimerization using my single molecule FRET approach and try and understand how dimerization lifetimes are changed as we alter lipid compositions. So definitely, I think it's very important. Again, very understudied, but something I'm very interested in doing as well.
Fantastic. And I think that's about the time we have for questions. Is that right? Should we move to the next talk? Okay, great. Well, thank you. Thank you so much again. That was fantastic. Next up, we will be hearing from Siobhan Meltzer, who is a neuroscientist at Harvard Medical School. We have a bit of a theme in this session, and she studies somatic sensation. So thank you so much, Dr. Meltzer, and I will turn it over to you.
Thank you for the kind introduction. And my name is Siobhan Meltzer, and I'm a postdoc in David Ginty's lab at Harvard Medical School. And I'm excited to talk about my postdoc work on the molecular and cellular mechanisms of mammalian touch circuit assembly. So we know that neurons in our nervous system use axons to send information and dendrites to receive information. So the precise forms and connectivities of axons and dendrites are critical for the function of the nervous system. And axons and dendrites also mainly communicate using synapses so that information flow from the presynaptic terminals to the postsynaptic terminals. So when I first saw these beautiful drawings made by father of modern neuroscience, Raymond E. Cajal, in college, I was struck by the desire to better understand how such diversity of form and therefore function arise during development. Specifically, I'm interested in understanding two questions. What are the organizational principles of the nervous system, and what molecules instruct the precise assembly of the nervous system? I think understanding these questions will not only bring new understandings and concepts in neurobiology, but also provides new molecular and cellular targets for treating neurological diseases. So we know that among all of our senses, touch is one of our basic senses, but it's really least understood at molecular and cellular levels. We rely on our touch sensation for our physical interaction with the world and also for social interactions. So therefore, precise and proper level of touch sensation is really important for our daily life. So, however, coming from a neurodevelopment background, I realized that we really had very little understanding about how we develop our sense of touch. And at that time, David Ginty's lab really developed a lot of mouse genetic tools to allow us to get early and specific genetic access to different types of touch sensory neurons in mice. So this is when I decided to take advantage of these new tools to establish a niche in understanding touch sensation development. So past work from our lab and others has made significant progress in understanding the mature circuits that mediate our sense of touch. The first step in any somatosensory perception involves the activation of primary sensory neurons whose cell bodies reside within the dorsal root ganglion, or DRG. And the subtypes that detect touch sensation is called low threshold mechanoreceptors, or LTMRs. There are many different types of LTMRs. They all send a primary major axon arbor to the periphery, where they further branches out to form different types of mechanosensory end organs to detect different types of mechanical stimuli. And we believe that the precise and unique structures for each subtype is really important for their unique response properties to different types of mechanical stimuli, such as hair deflection, indentation, and a vibration. On the other end, LTMRs send touch information to the spinal cord, and here they form synaptic connections with spinal cord neurons to relay touch information. Here, these axons terminate in distinct but overlapping regions of the spinal cord. And from there, touch information is further relayed to the cortex for touch perception. As you can see, although we know a lot about the mature circuit that detects our or mediates our sense of touch, we actually know very little about how the circuits are established during development. We have some general ideas about their developmental steps because the touch sensory neurons share similar developmental milestones as other types of somatosensory neurons. So we know that in mice, these neurons are born at around E10. They migrate and form ganglion, and they go through a process of neurogenesis and transcriptional maturation. And they form these two major axon branches to go to the periphery and to the spinal cord. Among all of these processes, I would say that we know the least about the later developmental steps, such as how they form unique and specific mechanosensory end organ structures in the skin, and how they form specific connections in the spinal cord to relay touch information. And these are not just the basic questions about touch circuit development. These are also presumably the steps we want to correct or reenact in order to treat nerve injuries and somatosensory disorders in neurological diseases. So with these questions in mind, my postdoc work addressed or investigated the molecular mechanisms of touch circuit wiring. Specifically, I did this in 3 papers or projects. First, I characterized the development and patterning of touch sensory neuron peripheral and central projections. I did an RNA sequencing to identify candidate signaling proteins that regulate touch sensory neuron development. From a list of candidate genes, I published 2 papers on the molecular pathways for touch sensory neuron development. One is mainly involved in regulating the formation of the sensory end organs in the skin. And another story is mainly about how protocadherin can regulate synapse formation and peripheral branching of touch sensory neurons. For the interest of time, today I'll give you a highlight about my latest story about the role of protocadherin gamma genes in touch development. So the protocadherins are predominantly expressed in the nervous system. They constitute the largest subfamily of the cadherin superfamily of cell adhesion molecules. The cluster of protocadherins are all reside in the same locus in the mammalian genome. The cluster genes comprises alpha, beta, and gamma subclusters. For the alpha and the gamma clusters, the isoforms are assembled by splicing of one variable axon and transmembrane domains to three constant axons encoding the constant shared intracellular domain. As a result, each isoform or each gene encodes a specific protein isoform with unique extracellular domain, unique intracellular domain, and they all share a similar intracellular domain. So we know that the cluster of protocadherins are important genes for neurodevelopment because mutations of these genes are associated with neurodevelopmental disorders such as autism. So the protocadherin cluster genes are really play many roles in neurodevelopment, and one of their most famous roles is their role in dendritic self-avoidance. So in the mouse retina, if we remove all the 22 protocadherin gamma genes in the retina neurons, their dendrites don't spread out evenly anymore, and they form these bundles. So this is proposed to be mediated by their roles in repulsion. So in mediating repulsion between dendrites of the same neuron. And protein structure work also identify the exact mechanism of their homophilic binding mechanism. So each isoform only binds to the exact same isoform on the other side of the membrane. And together they form this lattice structure to activate downstream signals. But for today's talk, I'll tell you about a new role for the protocadherin gamma genes that's rather adhesive and not really repulsive. So from my RNA sequencing analysis, I saw that many genes at the protocadherin gamma locus are expressed at different levels in developing touch sensory neurons. So we obtained the FLOX allele in which the last common exon is FLOX, and there's also a GFP fusion here to confirm the deletion. So we combined this FLOX allele with sensory neuron-specific Cre, advalin Cre, to generate sensory-specific conditional knockout animals in which only the DRG are missing the green protocadherin gamma genes. So we did a number of behavior assays first with these conditional knockout mice. And we found that the animals were all did really bad for all of these touch sensation assays. They're really bad at sensing and reacting to touch stimuli applied on their skin. To further look at their cellular loci of dysfunction, I looked into the spinal cord where the touch sensory axons come in and they form synaptic connections with the spinal cord neurons. Here, I found that there's one specific deficit in synapse formation. So we could visualize these touch sensory axon terminals using these synaptic marker in magenta. And we could also visualize the synapses they make by staining for the excitatory postsynaptic marker HOMR1. And we could overlay these two channels to specifically look at synapses formed by the sensory terminals. Here we found that although there's no change in the density of sensory terminals, each sensory axon terminal forms fewer and smaller synaptic connections with the spinal cord neurons. We also looked into the skin to see how they form these sensory axon pattern in the skin. Here we found that in the sensory conditional knockout mice, each neuron form fewer branches in the skin, and they innervate fewer hair follicles, which is highlighted by these red circles. Interestingly, when we knock out the protocadherin gamma genes altogether in the magenta terminal Schwann cells that are closely associated with these developing axons, we found that these Schwann cell conditional knockout animals also showed similar deficit in sensory axon branching. Together, these experiments suggest that the protocadherin gamma genes can mediate the interaction between the Schwann cells and the sensory axons to promote axon branching. So just a quick summary, we found that there's different roles for the protocadherin gamma cluster gene on the two different ends of the touch sensory neurons. So in the periphery, they can promote axon branching through mediating interactions with the glia cells. In the spinal cord, they can promote the formation of excitatory synapses between the touch sensory neurons and the spinal cord neurons. And for the interest of time, I'm summarizing another important finding here. We used a combination of mouse genetics and also in vitro artificial synapse formation assay. And we found that C3 is the only isoform out of the 22 isoforms here that's really important for promoting synapse formation. It is both necessary and sufficient for promoting synapse formation. So using these sensory conditional knockout mice as a model, we further investigated whether the mechanosensory inputs to the spinal cord is important for the spinal cord circuit function and assembly. Specifically, we looked at the innervation of the spinal cord by the cortical spinal tract. So normally in the spinal cord dorsal region, they receive descending input from the cortical neurons. So their cell bodies reside in the cortex, and then they send a long axon down to the spinal cord to form synaptic connections. And we can visualize these synapses by injecting virus into the cortex. So we found that in the sensory conditional knockout mice, there's a reduction of the density of these magenta cortical spinal terminals. And at the same time, each cortical spinal terminal forms fewer synaptic connections with the spinal cord neurons. In addition to this assembly changes, we also found a number of spinal cord neuron function changes in these animals. So as a summary for this project, we found that the cluster protocadherin isoform diversity is very important for touch sensory neuron synapse formation and peripheral axonal branching. We're particularly excited about the direct role of C3 in initiating synapse formation. Many genes at the cluster protocadherin gamma locus are expressed broadly in the developing nervous system in a combinatorial manner. So we think that the direct role of C3 in synapse formation may allow this cluster gene to be a possible code for synaptic wiring specificity. For example, neurons that express the same or similar set of protocadherin gamma genes might preferentially connect with each other. We also found that mechanosensory inputs are important for the precise assembly of the somatosensory circuit. So my future lab will build on this framework to further investigate the molecular regulation and the coordinated assembly of the mammalian somatosensory circuit. I believe there are many open and underexplored questions about somatosensory circuit assembly. So as the first aim and also a short-term goal, I will further investigate the molecular mechanisms of touch sensory neuron peripheral and central innervation. And for my second aim, I will use a combination of 2-photon live imaging and mouse genetics to better understand the molecular and cellular mechanisms of touch circuit assembly during development and also after injury. For my third aim, which is a combination of short and long-term goal, I want to use the sensory neuron and the spinal cord neuron as a system to better understand how they coordinate their development to form a functional circuit during development. And for this aim, I plan to use a combination of spatial transcriptomics, mouse genetics, physiology, and behavior to further elucidate what are the molecular and cellular steps that allow the sensory neurons and the spinal cord neuron to wire together during development. My long-term outlook is to better understand how the somatosensory circuits goes awry in neurological diseases, which will provide molecular targets for treating somatosensory dysfunctions. So with that, I'd like to thank you for your attention. And I'd like to thank my postdoc advisor, David Ginty, and also my lab members, collaborators, and my funding sources. Thank you, and I'm happy to take questions.
Great. Thank you so much, Dr. Meltzer. We have a question here, which is, can you please tell us a little bit more about the 22 isoforms in terms of their expression pattern, cell or tissue specific, and also whether they have different functions?
Oh, yeah, that's a very interesting, very important question. So in the DRG neurons, we saw that the C3 is expressed at very high level in the developing touch sensory neurons. And similarly, in the spinal cord, C3 is also expressed in about 60 or 70% of the spinal cord neurons. So this is consistent with our model that C3 can promote synapse formation between the sensory neurons and the spinal cord neurons. And at the same time, we also looked at a few other representative isoforms from this locus. So in the DRG, most of the other isoforms are expressed in a small subset of neurons. And this is true also for the spinal cord neurons. Interestingly, there's also a new study showing that the C4 isoform from this locus playing important roles in promoting spinal cord neuron survival. So it looks at, so if you get rid of C4 isoform only in the animals, there's a lot of spinal cord neuronal death and the animals die at birth. And we don't yet know what the mechanism is for C4 in neuronal survival. And it's very interesting to me that many different isoforms at this locus could have very different roles. So to me, one hypothesis is that their unique intracellular region might be associated with different downstream signaling protein to activate different downstream events.
Great. And I think we have time for one more question. Does that sound okay? I can ask one. I was wondering about the stimulation of the hair follicles that you described. I was wondering if you know a little bit more about that cell-to-cell interaction or what compartment of the hair follicle is being affected in that model. It's quite interesting.
Yeah, that is a very interesting and very important question. How is mechanotransduction initiated around the hair follicle?
Yeah,
so
exactly.
yeah, so we know that for the types of touch sensory neurons that are really sensitive to deflection, they form these kind of fence shape structures around the hair follicle, and these they basically go parallel with the hair shaft, and this. We think that this structure really allow them to be very sensitive to bending of the hair. And interestingly, we also have a recent study on bioRxiv. Actually, it came out on Neuron recently. So we did a EM reconstruction of the mechanosensory end organ structures around the hair follicles. And we proposed a model about how the structure can facilitate detection of touch. And we also found that the mechanosensitive ion channel piezo2 are expressed in these sensory endings around hair follicles.
Wow, very interesting. And congratulations on all of the recent publications. That's fantastic.
Thank you.
Great. Thank you so much. And I think it is perfect timing to move to our next talk, which will be from Dr. Deborah Kamin-Mukaz, who's coming from the University of Vermont and working on the intersection between molecular and social epidemiology. So we're really switching gears. And great, I'll pass it along.
So thank you. So my name is Deborah Kamin-Mukaz, and today I'll talk about cardiovascular disparities and with a special focus on hypertension and the effect of thromboinflammation and structural racism. So this is just schematic of my career trajectory. Currently, I'm doing my postdoc at the University of Vermont, and I'm focusing on the molecular and social epidemiology of disparities in cardiovascular and cardiometabolic health and COVID-19. And to give you a cohesive story, I am focusing on 3 different projects, and they will make sense when we talk about my future goals. So let's start. So cardiovascular diseases are leading causes of mortality and morbidity globally and in the United States. And hypertension is an important cardiovascular risk factor. As you can see here, when it comes to the population attributable risk of hypertension for heart disease and stroke, it's quite high, it's quite elevated. And when you look at the American Heart Association metric for cardiovascular health, we see that they made sure to include blood pressure, again, because hypertension is an important cardiovascular risk factor. So the pathogenesis or development of hypertension is linked to the interplay between thrombosis and inflammation, and that is known as thromboinflammation. So when you look at this figure, under normal conditions, when there is injury to the vasculature, you don't see dysregulation or chronic vasoconstriction. But under conditions of endothelial dysfunction, you do have vasoconstriction. But what you also have here is dysregulated hemostasis or thrombosis and inflammation. And eventually, these dysregulated processes are going to impair blood flow and potentially lead to a high blood pressure or hypertension. And D-dimer is a key thromboinflammatory biomarker. So what is D-dimer? D-dimer does not have an enzymatic activity, but it is a byproduct of fibrin. And what is fibrin? Fibrin is the mesh that holds the blood clots together. And higher D-dimer reflects hypercoagulability, is associated with inflammation, is a predictor of cardiovascular risk. And with the pandemic, what we also have found out is that D-dimer is the best estimative biomarker of the thromboinflammation and poor outcomes during SARS-CoV-2 infection. So the biology underlying D-dimer is very important when it comes to thromboinflammation. And there are significant racial differences in D-dimer levels. So Black people have higher levels compared to white people, and Black people also have higher levels of other hemostatic and inflammatory biomarkers. Which brings me to my first project on the associations between thromboinflammatory biomarkers and D-dimer in a biracial cohort, and I'll talk more about the biracial cohort. So my research question was to know whether there were racial differences in association of those thromboinflammatory biomarkers with D-dimer. So first, I want to talk about race. I want to make something very clear. Race is a social construct as we are discussing these biological differences. In a lot of population studies, it is self-reported. It's supposed to represent bioancestry, but race is fluid. It depends on the sociopolitical, geographic, and economic context of the time. As such, in molecular epidemiology and genetic epidemiology studies, we are moving away from race as reflective of genetic ancestry. So our study was the reasons for geographic and racial differences in stroke study, or REGARD. So REGARD recruited more than 30,000 Black and white participants aged 45 and older from 2003 to 2007, and then did a follow-up examination from 2013 to 2016. And REGARD oversampled Black participants and oversampled participants in this southeastern region of the United States, also known as the stroke belt region of the US because of the higher rates of stroke. The objective of REGARD was to identify causes of racial and geographic differences in stroke mortality and cognitive impairment. And REGARD is richly phenotyped. So some of the data, we obtained a lot of data, including blood and urine samples that were shipped at the University of Vermont, where I am, for storage and analysis. So when it comes to the analysis for this particular study, as you can see here, a lot of these biomarkers, the majority of these biomarkers were associated with D-dimer. And these biomarkers in blue here showed significant racial differences. So for instance here, in the general population, D-dimer was 18% higher per each standard deviation higher, or 46% higher factor 8. And then when you look at the differences in the association by race, what you do see is that for those three biomarkers, we saw stronger association in Black people. So what we concluded is that in Black people, you have greater thromboinflammatory responses, which might partly explain their higher rate risk for cardiovascular conditions. But these findings were not explained by socioeconomic and clinical factors. So we adjusted for those factors, and we did not see an explanation for that. So we have to start looking higher. We have to start looking at structural racism and thromboinflammation. So for my second project, I looked at D-dimer and the risk of hypertension in regard again. So hypertension is more prevalent in Black Americans compared to most of the ethnic and racial groups here in the United States. And the majority of hypertensive cases are grouped into what is known as essential or primary hypertension. And the underlying causes of essential hypertension are still ill-defined. However, it's possible that thromboinflammation may play a role in the development of hypertension. And as I previously said, D-dimer is an important thromboinflammatory biomarker and is higher in Black people. So I had 3 questions. My first question was to know whether higher D-dimer was associated with a higher risk of hypertension, and whether the association differed by race, and whether D-dimer explained or mediated racial differences in the risk of incident hypertension. So I showed that higher D-dimer is associated with a higher risk of incident hypertension. So when we compare the 4th quartile of D-dimer versus the 1st quartile of D-dimer, we saw that the risk was 22% higher. However, the association did not differ by race, and D-dimer did not mediate or explain racial differences in risk of incident hypertension. So we concluded that the thromboinflammatory mechanisms underlying D-dimer variability may play a role in the development of hypertension. And then when it comes to future research, we have to start thinking about the biological pathways linking D-dimer, hypertension, and hypertension risk factors. So structural racism again, and thromboinflammation and its association with thromboinflammatory biomarkers is also going to be something that we need to look at. So the third project that I have here is on residential segregation and risk of hypertension in regard. And I received a postdoctoral fellowship from the American Heart Association. And my research is on residential segregation, thromboinflammation, and risk of hypertension. The residential segregation and the thromboinflammation analysis is ongoing. However, today, I'm going to talk about residential segregation and risk of hypertension. I want to make something clear again. Structural factors are drivers of racial inequities in hypertension risk and other cardiovascular diseases. And this book really highlights this problem, the color of law. When it comes to what is residential segregation? Really, residential segregation is the legacy of laws and policies that forbade Black people from living in the same areas as white people. And what residential segregation did was to take away resources and opportunities for Black communities. As such, it is a geospatial manifestation of structural racism. There are different aspects or dimensions to residential segregation. And for the sake of this talk, I'm going to focus on the evenness and the exposure domain of residential segregation. The evenness domain is looking at the uniformity of the distribution, and the exposure is looking at contact. So my research question was to look at whether residential segregation has a differential association with hypertension risk in Black and white adults. So in order to capture these dimensions of aspect, you have different indices. One of the index to capture the evenness domain is the dissimilarity index, which is really looking at the difference in race distribution of the census tract, which is the smaller subunits relative to the county. The isolation index is really looking at the degree to which Black people are exposed to only one another. And the interaction is obviously the degree to which Black people and white people interact with each other. So I want to be very careful when I'm saying this. When it came to this analysis, these indices were not associated with the risk of hypertension in either Black or white people. But we have to talk about the limitations. First, residential segregation is a geospatial manifestation of structural racism. As such, but these indices do not really capture that spatiality. So we have a limitation there. Then the second issue is the modifiable areal unit problem. If we were to change the scale or if we were to, let's say, to measure residential segregation at a state level or a neighborhood level, potentially these results might change. And third, our cohort was limited to Black and white adults over a certain age. So we have to remember that. So we concluded that these 3 measures were not associated with hypertension risk in Black and white participants of the regards, but this differs from another study that shows that a measure of racial clustering Racial clustering is really looking at the extent to which areas where Black people live adjoin one another, and that was associated with a higher risk of hypertension. So in conclusion, what we can say is that different measures or aspects of residential segregation capture factors related differently to hypertension development. And for future direction, and I'm working on that, is really to look at these other domains of residential segregation and use spatial measures for them. So what's next? So I am training as a molecular. The goal of my career really is to look at the molecular and social epidemiology of cardiovascular and cardiometabolic inequities in historically marginalized communities. So for the social epidemiology piece of it, I want to look at the impact of systemic oppression on cardiovascular inequities. And for the molecular epidemiology piece of it, I want to look at the biological embodiment of systemic oppression. So I'm going to use an omics approach to do that. And as a first step, in June, I submitted a K-99 on the intersectional impact of structural racism, structural sexism, and structural classism on cardiovascular health and cardiovascular CVH-associated thromboinflammation over the life course. A very long title. The other thing is that because of the topic of my work, I'm very passionate about DEI and advocating for researcher, and I want to include that within my career. I'm the current president of Black in Cardio, which is an association or an organization to celebrate Black people in cardiovascular fields and raise awareness about cardiovascular diseases in Black community. And I plan on continuing on doing my work in Black in Cardio. I want to thank my co-authors and all the different founders. I want to thank my mentor, Dr. Mary Cushman. And if you have any questions, this is the time to ask.
Great, thank you very much. That was so eye-opening. Thank you for your talk. Everyone can just add their questions to the chat, and I'll start with one now. I've never seen someone working at such a two very different ends of the spectrum in terms of your research. It's so impressive. I was wondering where it starts for you in terms of, do you do field work to collect the data regarding, for example, the inequities and racial segregation in residential arrangements, for example, that you were talking about? How do you get that data or analyze it? What does that look like on your end?
So these are census data, because when it comes, and it's a larger conversation, there are different dimensions to oppression. You have the interpersonal type of oppression, then you have the more structural type of oppression. And when it comes to structural, you're looking at laws and policies. How do you know about laws and policies? You look at census data, you look at locations, you look at the number, when it comes to residential segregation, just kind of the conformation of where people live and how they live with each other. So census data is very important when it comes to measuring these different measures.
I see. And does the quality of that data vary? Is that part of your kind of critique? I could imagine there might be some variability.
The quality, I think the question really is, you might think about it as a bigger philosophical question. It's the quality of the data is very important. And also, what does it mean when we talk about census tract? These are administrative definitions of where people live, but people don't necessarily interact with each other within a census tract. When I think about my neighborhood, I don't necessarily think about my county. I think about, there are different ways to look at space that is different from what we have when it comes to administrative data. And you remember I mentioned the modifiable aerial unit problem that is going to play a role in that. So, but it also speaks to the fact that we are trying to measure these very highly complicated sociological phenomena. So we have to start understanding the limitations of what quantitative analysis can do.
That makes a lot of sense. Thank you. Great. Okay, the next question is, when considering residential segregation, how much does access to healthy dietary sources affect cardiovascular health? Is there a way to tackle this challenge in areas that might be considered food deserts?
Yeah, because when it comes to residential, it's when it comes to residential segregation, it's really about taking away resources from Black communities.
Mhm.
So in areas that are majorly Black, there is, we have to also be careful when we think about how people live with each other, because there is also this issue with gentrification, where you might have, the problem is not necessarily that people want to live with each other. It's what it means for Black people to live in an area, because areas that are majorly Black, what you see is that resources are literally being taken away. Banks are taking away resources. And there was a recent analysis that really mapped stores, grocery stores, and just the segregation in the United States. And it's very clear. Food deserts, residentially segregated areas with a majority of Black people. So that's where it gets complicated. It's really what it means. It's really about taking away resources and opportunities.
Thank you. The next question is, studies have found a link between discrimination and the gut microbiome. Do you think there might be a link to cardiovascular health? And are you planning to look at that in your future work?
I haven't thought about the gut microbiome, but I also have to kind of explain how systems of oppression work. So when it comes to discrimination, the first question I would have to ask is, what type of discrimination? Are we talking about personal discrimination? Are we talking about the most structural type of discrimination? Because when you look at personal discrimination, I guess, which is what most of the studies do, is the prejudice, the personal prejudice that people have toward one another. But when we look at structural oppression or structural discrimination, what we are looking at how that personal discrimination gets translated in laws and policies and rules, and who has access to power and all that. And I don't know if something like that has ever been done, because I'm more interested in the structural part. Obviously, I want to look at how the structural oppression interact with interpersonal oppression, but my focus is really about the structural piece of it.
Great. That's helpful. And I think, do we have time for one more question? Yeah, one more. Okay, this will be the last question. And thank you for all your answers so far. What omics approaches do you plan to use in your future research to look at the biological factors? For example, genes, RNA, proteomics, metabolomics, et cetera.
Yeah.
And is anyone better than the other?
I don't think that's the thing. It's like the field, to be honest, not a lot of has been done when it comes to just large data sets and looking at just that biological translation.
Mhm.
Right now, I'm focusing on proteomics, but what I'm going to do is I want to also look at the interaction between with genes, because the conversation now is really looking at how these systems are changing the genome and how that's interact with biomarkers within our body. So eventually I'm going to move to other types of omics, but right now it's proteomics.
Great. Well, I can't wait to follow your work in that direction. And thank you so much for the talk.
Thank you.
Okay, great. So now our final talk of the session is from Dr. Luis Hernandez Nunez, who is a neuroscientist at Harvard University, who's working on heart-brain connections and a number of other fascinating topics. I'll let him tell you about it. So thank you very much.
Thank you. Thank you for the introduction. Yes. So I mean, my interest in science started with, in academic research started with control. And when we think about control and control systems and what they do, what immediately comes to mind in the field of biology is homeostasis, and it's true. Control mechanisms are an essential piece of living organisms, and they're present from a bacteria that needs to regulate protein concentrations, all the way to the biggest mammal, to a blue whale that needs to regulate heart rate, and to everything in between. And actually, there are only two types of systems that need control mechanisms. One is biological systems, and two is man-made systems. We've learned from biology to engineer machines and systems like the internet or like a plane that use a variety of control mechanisms. So the way I started thinking about control and feedback control in particular was when I was an undergrad, very early in my undergrad studies in mechanical engineering. When back in Peru, I'm Peruvian, I worked in the project of the first Peruvian satellite launch into space, and we had to find a way to control the orientation of the satellite after it's launched. And in doing that, I got to learn a lot about control theory, and I came across a lot of literature in biology, which was fascinating for me because I had learned and always thought of control systems as things implemented in code you write and in electronic circuits. And it turned out that you could implement very robust, very efficient control systems in something as simple as a bacterium using molecular signaling pathways. So still as an undergrad, I had the opportunity to move to Yale University, and in Thierry Monet's lab, I studied how do bacteria keep proper coupling between chemoreceptors and flagellum motors? How do those proteins stay in a functional range? Looking at this question of how bacteria behaves and their behavior is coordinated by a signaling pathway, and how the pathway controls the activity levels of the components, made me think of, okay, if this can be studied in a molecular network, what about a neural network? And for my PhD at Harvard, I switched to, I did a PhD in systems biology, but I worked in the neuroscience question. I worked on how fly larvae can regulate or use behavior to regulate body temperature. And I had to do things that first to figure out how does the animal detect the signals, how does it detect temperature. And one of the main findings of my PhD is that I discovered the molecule used for sensing warming, IR68A, is a thermoreceptor, and the co-receptors 93 and 25 that turned out to be useful for the warming cells that I found, and for the cooling cells that had already been discovered in the fly larvae. And after doing that, I wanted to know how the information of those sensors is used to control behavior, which results in regulating body temperature in the case of the larvae. And we use connectomics, calcium imaging, optogenetics, and behavioral analysis. And we found circuits that already implement components of a computation for control that I had predicted. And the success of this project, we're going from a molecule to how the control system is implemented in a neural circuit, motivated me to think of fundamental biological questions that need or have not been approached from a control theory perspective. And near the end of my PhD, I started reading about what other systems might benefit from this approach that have not been studied with it. And I was very surprised when I started reading about how organs are controlled. It's been 100 years since, a little more than 100 years since Langley divided the autonomic nervous system, the system that connects the central nervous system with organs to regulate them, in two. It divided them in sympathetic and parasympathetic or vagal systems. One of them, the sympathetic, is a fight or flight system. It's gonna mediate increases in heart rate. It's gonna mediate excitatory motor stimuli in the organs. And the parasympathetic is the opposite. It's a rest and digest one, at least that's the way it's described in medical text, and it sort of compensates. And the equilibrium between the two will regulate organ function, or at least that's the idea. But this qualitative description has been the core way of thinking about it, and there is a lot of computations that need to happen for this to take place. And in recent years, there's been a lot of interest in like the genetics of the receptors in the vagus nerve, and a newfound interest in the autonomic system. But I could not find labs that were approaching this from a control systems perspective or a systems neuroscience perspective. And actually, this is the way I think about the problem, and I think this is one of the ways it could be described. There are many circuits in the central nervous system. Some brain regions have been identified, but we don't know the dynamics of the circuits needed in the brain to then regulate the autonomic nervous system that then will regulate something that is even more understudied, the intra-organ nervous systems. So every organ, and sometimes we don't think about it in neuroscience, we're so focused on the brain, but every organ has a set of neurons that coordinates the function of the different cell types in the organ. For example, in the heart, the neurons are going to regulate or going to communicate with cardiomyocytes and pacemaker cells. And we don't really know what they do. We know very little about them. In the case of the heart, had to go back several decades to find any studies that had tried to study these neurons. And there's some basic experiments that are the minimum necessary to try to study this from a dynamical systems perspective. The first one is we need to be able to measure activity in individual cells, and ideally in more than one part of the system at the same time. So we don't have to average across animals, which sometimes when you have to average across animals, you don't have enough information about each system, and you lose so much in the average, and that you might never figure out how the control system actually works. We have to perturb the function of the cells so we can validate what we may hypothesize they do based on activity. And then with this information, we can refine our models of what we think might be implemented in a control computation. So to be able to do this, for example, to measure interoception in mouse, to measure the vagus nerve, you need to anesthetize the mouse. So I wanted to do this in a behaving animal, and also I wanted to be able to have access to ideally autonomic and central nervous system at the same time. So the only genetic model organism, vertebrate model organism where this is possible is zebrafish. So this is the star of the show. And in particular, the zebrafish heart. I decided to start with the zebrafish heart because you can study the state of the heart by looking at it mechanically. And because the zebrafish is transparent, you can see the heart from the outside without any intervention. So same as in mammals, in zebrafish, there's a sympathetic innervation of the heart. There is a motor vagus or parasympathetic innervation of the heart. And there's a sensory vagus, which is the pathway that sends sensory information from the heart and other viscera to the brain and then modulates brain function. But this is known in the adult zebrafish. And what actually made zebrafish widely used in neuroscience is that the larva is transparent. So it's very important that it's a larva, because then you can have optical access to the brain to record whole brain imaging and all the experiments that have made zebrafish strong systems neuroscience organism. But the autonomic nervous system had not been studied in fish at that point. And so we had to do the groundwork to find ways to study it and see if it's developed. So the first question we had to address is whether there's a connection between the heart. The heart in the fish only has one atrium and one ventricle, and because they don't have lungs, so they only need one pump to distribute blood to the body, and the brain. And the question is relevant because it didn't necessarily have to be the case. Zebrafish larvae can survive several hours without heart function. So it was very important to make sure there was neuroregulation. And to do that, the first thing I did was to build a simple setup where we could measure what the heart is doing using infrared light, and we could use traditional virtual reality environments in fish to show visual stimuli, to present visual information. And this is a high throughput setup because we don't need fluorescence in the heart to measure what it's doing. We can estimate from infrared illumination and how much light goes through atrium and ventricle, whether they are contracted or dilated. And we can use stimulus like looming. Looming is a visual stimulus that simulates the approach of a predator. So the fish thinks it's going to be eaten and it gets really scared and the heart rate goes up, and then it will trigger escape response. This is just one example of what it looks like. So each of these bars is a moment of looming, and then the fish gets scared and the heart rate increases, and the tail flick also increases. The tail flick means the fish is trying to escape. Although there is no real predator, but it's trying to escape because it's scared. So we studied first what's the first developmental point where a visual stimulus can be turned into a heart rate response. And we found that that's 5 days. So at 5 days post-fertilization, 5 days of age of the zebrafish is the first stage at which there is a response. Behavioral responses can occur at 4 days and 3 days, but not at those ages. There was no cardiac response. Then the response will amplify, and the dynamics will be more stereotypic as the fish gets older. This is a fish at 12 days. And simultaneously, we also stained our fish hearts. So this is ventricle and atrium. So we're staining for cardiomyocytes in magenta, and for neurites with acetylated alpha tubulin in cyan. We could also see that denervation is progressively appears first at 5 days, coincidentally with onset of heart rate increases, and then it will get to more heart regions and eventually to the valve, to the atrioventricular plexus, which is a valve between the atrium and ventricle. This was, of course, just the beginning, and then the question is, this innervation, where is it coming from? Is it from the sympathetic system? Is it the parasympathetic? Is it sensory? Is it all of them? And if so, which ones are doing what? So to do that, me and my team of scientists, we've done a lot of work in each of the branches, and I'm going to show you a very brief summary of our findings. So the first branch of the autonomic system that innervates the heart is in fish is the motor vagus, or the parasympathetic system. And in this work, Morgan and Sky have done a lot of it. And we used a variety of pharmacology, optogenetics, laser ablations to show when does it become functional. One of the anatomical results that was what set us on the path of studying that was that we looked at what's the first moment where the motor vagus innervates the heart. This is a tune of the zebrafish heart. And this is our summary. We found that at 5 days is the first moment where the motor vagus innervates the heart. But there are no heart neurons yet. The first heart neuron that we call the pioneer neuron appears on day 7. And then more neurons will appear. And at the bottom, I'm just showing you confocal images of the neurites in the heart. And with a red arrow here, it's just notable new traits that appear at each age. Then we did a lot of functional studies to verify what goes on. And basically what our conclusion was that at 4 days, the heart is basically not connected. It's just operating at a constant pace. And at 5 days, the motor vagus will regulate the heart at 5 days. And it's really interesting because you wouldn't expect that the motor vagus is sufficient to trigger a heart rate increase in response to a threat, something usually associated with fight or flight. And we've done similar studies, again, using calcium imaging, optogenetics, and pharmacology that I can discuss later. So for the sympathetic system, Areni and Kalista figured out that sympathetic system innervates the heart later. It appears at day 7 and it's already functional at day 7. So we go from day 5 where there's only motor vagus control to day 7 where there is motor and sympathetic control of the heart. And similarly, in my team, Joanna and Annette have been working on the vagus system, and we found that this is the working model that we have of how the system works. At 12 days is that the feedback from the motor vagus becomes useful to update heart rate. And this is really beautiful because it means not only that we can study our question in larval zebrafish, but also that we have a lot of mechanisms for developmental controls, depending on when each of the pathways becomes functional in the case of the heart. And this is just the beginning. What we're doing now, and what's going to be also continue once I become an assistant professor, is that we have developed a system to do brain imaging simultaneously with cardiac monitoring. So similar to the initial setup that is well known now for whole brain imaging in fish, for functional whole brain imaging, we've added to that design the capability of tracking the heart. And we're studying in different developmental contexts. So depending on how the autonomic system is, we study how the brain dynamics are being shaped by that to either control signals, or when it starts getting feedback, how is it modulated by interoceptive feedback. And similarly, we've developed a microscopy setup to track heart neurons. The heart neurons have been very challenging to study in the past because they move when the heart beats. So you have to really get a really fast volume and track them as they're moving to get their activity. And we have a setup to do this. And we're also studying it through development from the moment where there's only motor vagus all the way up to when feedback control is implemented with interoceptive function. And of course, this is just the beginning. The fundamental question is that for organismal homeostasis, multiple organs need to be controlled, not just the heart, and they have to be coordinated. And in the long term, I hope to study more organs. For example, the gills could be the beginning, to try to figure out how neural circuits will assemble during development to be able to control how these organs work and how also these organs will modulate the brain to affect behavior and potentially cognitive function. None of this work would have been possible without my amazing team of postdocs, master students, and undergrads, and without Harvard and my mentors' goodwill to provide me with space and resources, and also without my funders that I have. These are my main funders, and thanks to that, I'm able to have a team of students. Of course, I'm also grateful to the Engert and Fishman labs. It's been ideal to work at the intersection of a cardiology mentor and neuroscience mentor working on this new project of understanding how neural circuits control organ function. Yes, and I'll be happy to take any questions.
Great, thank you very much. Fantastic talk. Aishwarya, are there any questions to start with?
Not really, but I was just going to ask something myself. So you talked about how you've worked a lot on zebrafish. I was wondering if you think there could be comparisons we could make to larvae of animals that have different life stages. So there are some stages of life that are in water versus when they become adults, they eclose like mosquitoes. You have larval stages in water and adults that are aerial. So how do you think the system would function? It would be a lot more complex, I'm guessing.
That's so interesting. So you mean for an insect that will change the way it behaves? Oh, usually I get asked the other way around, like how would it work for a mouse? And it's easier because actually it's very similar between the fish and the mouse. So I don't know how similar it is between fish and larvae, but so when I was deciding what organism to use, I worked in the fly larvae before working in fish. So I searched the literature to see, are the organs in the fly larvae connected to the fly larvae brain? And I couldn't find anything, and actually found information that maybe the rudimentary part of the fly larvae is not really connected. So it's possible that a lot of that control happens with humoral influences, with circulating influences, right? Through hemolymph, diffusion of small molecules and hormones. So that those mechanisms, I think, are still very interesting to study, even though there might be less similar to mammals still, because it's like an alternative way to implement a solution to a problem, and we could learn a lot from that. Yeah.
Very, very interesting. Thanks so much.
I had a question too. So at the 4 or 5 day time point, I think you said in the heart where you first start to see the neural connection of the heart. Do you know what part of the organ seems to the neurons come in contact with first, or how does that?
Yeah.
Is there sort of a specific regional organization to that?
Thank you. That's an excellent question. So yes, the first neuron that migrates to the heart goes to a region called the sinoatrial plexus, which is where the pacemaker cells are located. And something that we found that I actually, because of the length of the talk, it didn't make sense to show, is that it migrates to a region where pacemaker cells are, and the first onset is of heart rate control, like the main variable that I showed, and not of, for example, contractility. Like the force of contraction is not regulated yet. That will happen later in development. Yes, so studying the function of those neurons is really interesting. It has a lot to do with what the animal needs first, I guess. Yes, and it's the same for when the atrioventricular plexus, which is the other important part of the heart, gets neurons in. Also, new functions emerge, and then we're doing single cell ablations of those cardiac neurons to show what what each neuron does at each point in development.
Fantastic. Thank you for explaining that. Great. Aishwarya, what was that? Did you say it's time's up?
We're almost.
Okay. Great. Well, thank you so much for that talk, and to all of our fellows. This has really been a fantastic session. We really appreciate everyone coming out and sharing their work, and it's really been our pleasure to moderate today. And thank you also to all of the attendees who have been here with us. And so we will now take a 15-minute break, and I hope everyone will come back in a few minutes to hear our second keynote presentation, which is titled A Career in Balance by Dr. Huda Zogby. It should be a fantastic talk, so I hope everyone comes back for that soon. And great, with that, thank you very much, and I hope everyone has a wonderful day.
Thank you. Thank you, Rachel.
Thank you, Rachel.
Thank you, everyone.